The next presenter that we've got up is Dr. Ryan Hansen. Ryan is an assistant professor in chemical engineering. And Ryan, if you want to make your presence known on the screen here.
Yeah, I don't know if can you hear me okay?
I can hear you just great. Thank you.
Great. Okay, so
okay. Yep,
okay.
Floor is yours.
Okay. All right. Well, thank you, everybody that's tuned in today. It's certainly exciting for me to present some of the technologies that my lab has developed over the past few years. Certainly complementary, I think, to some of the sequencing technology that is being developed. I'm going to talk today about one specific technology called what we've termed the Microwell Recovery Array. This is a device that has been designed to rapidly explore microbiomes and specifically explore cell-cell interactions between different species that exist in microbiomes. So it's a lab-on-a-chip type system. If you're familiar with microfluidic devices or micro nanoscale technologies, this is an example of that as a new tool for screening and discovery. Next slide, please. So I won't go into too much detail with this because I think this audience appreciates this. But of course, we've moved from just thinking about microbes here about a bacterium in terms of their internal machinery to thinking about how microbes interact with each other in very complex communities. Go ahead and forward. And so I think this makes that point. In many microbiomes, of course, what you're going to see is not just a single organism, but potentially hundreds, thousands of organisms that are interacting with each other, likely in a biofilm, and forming interactions, right? And microbes are social organisms. And so we can think about symbiotic interactions, competition, antagonism, right? These are all organisms that are all interactions that we'd like to really develop a better understanding of, because if we can do that, not only are we gaining fundamental knowledge, but we're learning how to potentially construct communities for desired outcomes. And what we know, of course, is that these communities have a big say as to the health of the host, whether that's a plant, tree, or a crop, or your gut, or many other systems. And so, really, again, we're driven by the need for new tools, specifically for looking at interactions in these communities. Let's go ahead and forward. I think it's become well recognized that in this area and in the microbiome area, there are new experimental tools that are needed. And I'm specifically thinking now about cellular tools. And so looking at the cell-cell interactions that occur between live cells from a microbiome. And that's really what we're trying to get at with one of our approaches that I'm presenting on today. And so we're thinking about this with a lot of different applications in mind. These are the ones that we've had projects around or that we're planning on working on. Bioenergy, and I'll talk about this in a little more detail, but this is specifically a collaboration that we have where they're interested in growing poplar trees and understanding interactions that occur in the rhizobiome of poplar. Poplar is a very interesting and potentially useful bioenergy feedstock. And so there's quite a bit of interest in that and developing woody biomass through poplar. We're kicking off a project where we're looking at this with corn and food production and understanding interactions that are occurring in the root microbiome in corn. We've thought about doing this with drug discovery. And so if you think about looking at cell-cell interactions, right, where you might be inhibiting a pathogen from growing, or maybe you're looking at screening cells that could inhibit a cancer cell, right? So that's a cell-cell interaction that we'd like to be able to explore. And then, of course, gut microbiome, the animal microbiome are also potentially useful here. So anyways, you've been seeing some of the steps kind of progress here. So let me kind of start with the first column here. And so in all of these in all of these systems, of course, you're going to have a fairly complex microbiome where you have all these different organisms that are present. Our idea was basically to take a focal species, and so this is a species that someone cares about. This could be a pathogen, this could be a beneficial bacteria like a plant growth promoting bacteria. And we really want to find the important interactions that could be hidden in that microbiome that influence the function of our focal species. And so that's what we've designed this platform to do. And all it is it's simply a silicon device, and we use standard microfabrication techniques to drill holes into these silicon substrates. And so now we have tens of thousands of wells that are small. They're about 10 microns in diameter. And what that allows us to do, if you look at the assembly, the stochastic assembly process that's described here, is combine our test species, our focal species, if you will, with a random assortment of isolates. And we've been able to show that we can actually build a network. And so we can seed these cells in different combinations. Maybe it's 2 cells in a well on average, maybe it's 5, maybe it's 70. And we can control that to some degree. But the key point of this is that every well is a little bit different than the next in terms of the composition, in terms of the combination of cells that we put into these wells. And so that's where the screening comes in, is that we can look at these different co-cultures, if you will, and really scale up how much we can observe in a single test and potentially look at thousands of unique co-cultures. So when we do that, go ahead and forward. We basically use fluorescent time-lapse fluorescent microscopy that will allow us to track this focal species. Right now, we're just looking at its growth. And go ahead and play the movie. This is just an example of that. And so this is a time-lapse fluorescent image where we are tracking the growth of fluorescently labeled or expressing focal species. And what we look for is, if you see on your screen here, you should see an outlier that comes up. And this is an example of maybe we're looking at a pathogen and we find a well where the pathogen isn't growing so well. So it's being inhibited, possibly, presumably because there's other microbes there that are antagonizing it. So that's the idea. We've developed image analysis tools now to rapidly identify those wells that are outliers and statistically validate that. Each well has an address. And so now what we do, and this is really the key that makes this platform work, is we can actually expose different parts of this platform with light. And we've developed a membrane that's a photosensitive membrane. It falls apart under exposure to light. And so we basically can pattern light over these wells using a patterning tool in my lab called the Polygon 400. And that, go ahead and forward, that is the kind of, what that allows us to do is basically degrade a membrane, open up. It's just like opening up a can, opening up the top of that membrane so that we can then pull those cells exactly out of those wells. And we've looked at this process very closely and have really perfected that extraction process. And so we can go ahead and harvest those cells where we think we see an interesting interaction. Go ahead and forward. And pull those isolates out, recover them. Go ahead and forward again. And we can repeat that, so we can jump around from any well where we see something interesting. It might be growth promotion, it might be growth inhibition, but we can effectively recover those cells. And then what we do is we sequence them with 16S amplicon sequencing. And so that's the key idea behind the platform. There's a lot of information there, but hopefully that gives you an overview of what this technique can do. Really the most important point, go ahead and forward, I'll talk a little bit about some of the results that we've got specifically with the bioenergy application. So this comes in collaboration with Oak Ridge National Laboratory. It's a DOE funded lab that is really interested in poplar, again, for the bioenergy feedstock application. And they have many isolates that they're looking at. One of them is called Pantowea YR343. It's a plant growth promoting bacteria in this system. Go ahead and forward. And so what we've seen here is that you can do a monoculture with this. And again, this expresses GFP, so we can track its growth fluorescently. And so this is just a background culture. Go ahead and forward to the next part. And so what we see when we do a co-culture with root isolates is that we get a varied response. And effectively, you can see in the green box, we get growth amplification. In the red box, you can't see it as much on the fluorescent image, but we see a growth inhibition. And again, we have software that can identify this. So that gives us an address to extract from, and then effectively we can come in and pull out these cells and figure out who they are, and then validate that interaction in a secondary assay. And so these are a couple examples of some organisms that we've isolated that can either inhibit or improve the growth. Specifically, we've looked at some 5-membered multi-species consortia, if you will, that show growth promotion, which is exciting. Go ahead and forward to the final slide. And so I won't be able to unpack this in enough detail in the time I have, but what we do, just as a takeaway, is we'll do an off-chip analysis, because of course we want to validate that interaction in a more standardized assay. So we pull out a candidate in a screen, and then we do, if you will, a scaled up version of that in a 96 well plate format. And so we have our isolates, and in some way we co-culture those with our focal species. And so this is an example of the inhibition. And so the blue line is the control, where it's just a monoculture. The gray lines and the red line are the co-cultures, where we see that antagonism and are basically able to recapitulate that interaction. Go ahead and forward to the other part of this. And so on the other end, and this is really what we found very interesting, is that we've also seen that we can pull out multiple members from these wells. This was an example of a 5-membered community. And when we co-culture those together, we see this emergent effect, where we actually see growth promotion only when those 5 species are there. And if we take out those and look at them individually, you don't see that promotion. And so, that's really an emergent effect that we get from these multi-membered communities. So go ahead and forward. And so really what we're most excited about is the potential to not just look at single interactions, but consortia, multi-membered communities that potentially generate these emergent effects. And we believe that our platform might have potential with that. Certainly that's where we're interested in going, and this is fairly recent data. So, we don't know what's going on in this interaction, maybe from a metabolic level. We just know who's there. And so certainly we're investigating this more, but, certainly excited about the potential here. Go ahead and forward. And so that concludes my talk, and I should have some time for questions, but I'd like to acknowledge my group and the people that have worked with this, my grad students and my former postdoc, as well as my collaborators. So Dr. Tom Platt, assistant professor in biology, has worked very closely with me on developing this platform, as well as my collaborators at Oak Ridge and in industry. I put up a few papers that are either published or under review right now. And so those are at the bottom of your screen if you're interested in learning more, or maybe more importantly, certainly if you're interested in collaborating, please contact me. I put my email down here in the lower left-hand corner. So I love working with people that have interesting questions. I love working with microbiologists. I'm an engineer that develops technology, obviously, but certainly would be interested in unique questions that we might be able to answer with this screening approach. So that's all the time I have, I think, for my talk, but I certainly think I have a few minutes for questions. So
yeah, you absolutely do. And we do have a few questions. Excuse me, we do have a few questions here for you. One of them, any applications for fungal interactions, or does the well size need to increase?
Yeah, I'd love to do that. I don't know anything about how to culture fungi, but I see this is from Chris Little, and I know he does, I believe. Yeah, I'd love to do that. It's out of my expertise. I know it's going to require a redesign and making these wells bigger. We've thought about doing this with larger cells, with mammalian cells as maybe the host cell that you monitor, and then you're screening bacteria to see what influences like a cancer cell. Certainly we could do this with kind of looking at fungal interactions as well. It's just a matter of redesigning the wells. We can make the wells however big we need them to be. It's standard microfabrication.
Great. And then the follow-up question is there a potential to explore fungal bacterial interactions?
Yeah, I mean, I'd love to do that. There's potential there. I think from my perspective, I only work with bacteria. I don't know as much about fungal species and how to work with them, but I think the potential is certainly there. I think the same principles apply as far as this stochastic seeding, the recovery. It's the challenge with all of this is getting it's a culture-based technique. So finding the right culture media that will allow us to recover as much as possible, because of course we know that the majority of cells in these microbiomes are not culturable. And so finding that general culture media is a challenge, but I think certainly there are potentials here for doing this. And I'd love to do it.
And then this next question may be along the same lines. Will the microwell system will this microsystem be adaptable to mammalian cell culture methods to study host pathogen interaction? And what is the total volume of the wells?
Yeah, yeah, absolutely. So we've thought about this with screening host cells that are cancer cells and then screening soil for anti-cancer bacteria. Certainly, you could do that with the same principles. That I just need people that understand how to work with these different cells, because I work with bacteria and that's it. But yeah, it's a matter of redesigning these and having someone that understands how to culture and keep different mammalian cells happy. Total volume of these wells, it's about picoliter. So picoliter, and so in terms of dimensions, it's typically we have 5 micron wells, 10 micron wells, and they're about 20 microns deep. And so I think when you do that calculation, it's about picoliter in volume. So fairly small.
Okay, great. Yeah. And the last question we've got here is, would you be able to apply this to anaerobes?
Yeah, absolutely. So that's, we can vary, we have a live cell chamber in our lab, and so we can vary the environment there, so we can make an anaerobic, aerobic. So yeah, we can knock out oxygen and potentially do an anaerobic culture. And in fact, in some of my other projects, we're looking at anaerobic systems in anaerobic wastewater treatment systems, and so we're developing that capability already. So yeah, certainly good questions here.
Very good questions. Thank you so much. Is there anything else? We've got about a half a minute left. We can move on. That was great. Thank you so much, Ryan, for that presentation.
My pleasure.
As we move on through the university applications that we've got going here, the next presenter is Dr. Chris Little, and he is a professor of plant pathology. Chris?
Unmute. There we go.
There we go.
Yeah. So I want to move to a little bit of a different tack here, looking at fungi. And really an application of these kinds of approaches of the Illumina sequencing, looking at microbiomes. And so really this work was done by Dr. Rodrigo Pedroso, who was a PhD student in my lab. And so I'm sort of an innocent bystander, but I'll do my best there. And we're funded by various soybean commissions who are interested in seed-borne pathogens, pathogens of soybean, and the various microorganisms that can cause problems. So go ahead. So just to spend a couple minutes here at first kind of talking about, what a microbiome is. And a general definition is it's the fungal community that's within or on an organism, but I also extend that to maybe within the soil, so beyond just what's in or on an organism, but what's in an environment even as well. And so here's an example we can all relate to with the human. You have a large number of fungal species that have been associated, for example, with the oral cavity, less so in other organs. And so, work is going on with the human microbiome, and some work has shown that disturbances in the human microbiome lead to things like hepatitis or irritable bowel syndrome, obesity, things of that nature. Next slide. Likewise, plants have microbiomes, obviously. And so here are a couple studies that show the microbiome on cereal leaves, including wheat, barley, oat, rye, here in the top left. That's the phylosphere microbiome. Lots of examples of pathogens and necrotrophs or saprophytes and yeasts there. And in the bottom there, a study done in Arabidopsis roots that finds a large amount of ascomycetes, which are the predominant fungal group that's found in most plant-associated studies. So the question for us was, what about the seed microbiome? We're very interested in seed pathology and seed mycoflora. So we're interested to kind of learn about what's inside the seed. Next slide. So you can see kind of an example here of sort of the concept of a seed microbiome. And really, you can separate the seed microbiome into core microbiome and peripheral microbiome. That is, you know, whether or not the seed, as in the picture of A there, or B is healthy, or a green seed, or C and D. Those are pictures of damaged seed, or E and F. Those are pictures of disease seed. You know, regardless of what environmental conditions, storage conditions, what the host is, or what the composition of the seed is, you're going to have a certain set of fungi that are associated with the seed no matter what, versus the peripheral microbiome, which changes based upon different sorts of conditions. Okay, so the seed quality is really dependent upon who's there and what they're doing. So next slide. So we sort of went into this kind of project with a couple general hypotheses. First being, you know, can we identify and amplify fungal specific sequences from whole seed, whole soybean seed DNA? And then the relative abundance, prevalence, and incidence of seed-borne fungal operational taxonomic units, or OTUs, which I'll mention again later, is sort of different between samples, seeds, seed tissues. So really, I'm going to spend most of my time sort of thinking about this first general hypothesis. Next slide. And so you'll see lots of flowcharts like this today, I think, of a relatively standard kind of data acquisition and analysis process, where we take whole seed samples and we extracted whole DNA, so it's a mixture of both plant and fungal DNA, and then the amplification step to generate the amplicons of our microbiome community. We looked at the 18S RNA in this case, 18S DNA of the ribosomal DNA in this case, and did an initial amplification and then a nested amplification that was barcoded and then will be sequenced through the next generation sequencing platform. So Illumina was the approach that we used. And my student went to a workshop and also used an open source MySeq analysis software called Mother, which was used to generate contigs and remove sort of the unusable sequences, and then could align the usable sequences that had 99% match with each other into OTUs, or the operational taxonomic units, which are basically equivalent to species. And then those OTUs can be identified taxonomically in different databases, usually suggest using multiple databases to do this. Unite is a database for fungal communities, and the NCBI is the gene bank, of course. And then you get your results from that. Next slide. So looking at just relative read abundance from all the samples that we processed from the soybean seeds, you know, again, the ascomycetes represent the vast majority of fungal genera. And then we see some basidiomycetes, which are the same group as mushrooms, and zygomycetes, kind of like the bread mold you might see in your refrigerator. But ascomycetes make up lots of different pathogens, lots of endophytes, lots of saprophytes that are out there. And here on the right side of the screen are the two most common genera that we found in the sequence information: Alternaria, which is very common saprophyte or pathogen, and also Fusarium, which we have a particular interest in my lab, which include species that are pathogens, species that are saprophytes, species that are endophytes. And we really spend a lot of time looking at Fusarium because of its role as plant pathogens in soybean. Next slide. So the top 3 OTUs, or operational taxonomic units, that we found in our seed samples were 3 species: Fusarium proliferatum, Fusarium thapsidum, and Fusarium acuminatum. And so I kind of show that in the gray box. Fusarium proliferatum is a pathogen, and I want you to see on the right side a rolled towel pathogenicity assay and also a pot-based pathogenicity assay, sort of compared to control. You can see how the seedlings are not as thick, not as vigorous. It damages germination. So we can isolate these same pathogens from the seed using a cultural approach on culture media, and also detect them using the metagenomic kind of approach as well. So they are definitely important pathogens and are of interest to us. Next slide. So that brings me back to that idea of comparing culturable approach to a metabarcoding or metagenomic type of approach. And so these are 2 sets of samples that were both plated out onto semi-selective medium and run through the DNA metabarcoding process. And what we certainly see is that when we do the plating, the traditional method of plating, which is how we used to traditionally get a microbiome or do a microflora analysis, we might plate seed samples out on media, and we're not able to always detect a pathogen of interest. So the last column there, the Fusarium pathogens that I've been talking about, in sample 1, sample 2, sample 3, sample 5, yes, but the rest of the samples, no, versus the DNA metabarcoding approach, we detected Fusarium in every single sample we've ever tested. The same was true with another fungus called Phomopsis right next door there, which we were able to detect in every single sample versus only partial detection when we used the traditional approach. So this gets at some interesting questions or ideas when it comes to diagnostics. Traditional cultural diagnostics versus using these kinds of high throughput sequencing technologies for diagnostics. What's the answer to the question you really want? And it turns out the seed companies don't really want the real answer to the question. That's another story. Next slide. So just a last experiment to talk about, looking at seed tissues. So actually manually dividing whole seed into seed coat, the cotyledon, and the embryo. And the embryo axis contains a root and leaves to be of sort of the infant plant, if you will, and looking at the distribution of fungal pathogens and fungi in general in those separate tissues. Next slide. And here's just a sort of a visualization of that, just looking at sort of different patterns of proportional read distributions, or the frequencies of fungal genera among those tissues. And what's really interesting is some species like Alternaria and Cercospora, when you compare that with the species of interest, which is Fusarium, that pattern is really different. So the preponderance of Fusarium reads in both the embryo axis and the seed coat much greater than that of the cotyledon. And that's what we find when we do the cultural analysis as well. So we're seeing some connection between, even though this is semi-quantitative at best, we're seeing some connection between the reads that we get from the metagenomic approach versus the traditional cultural approaches that we've used in the past. So just one more slide, last one. I just wanted to revisit that seed microbiome concept again, which is very interesting to me you know, when we go through and sample and look at incidence among a great number of samples, and I think this may be from 45 samples here, you know, how we define a core microbiome is species that always tend to be there, regardless of conditions, versus peripheral microbiome, which are very conditionally present. And so in blue here, you can see examples of some species that are always, OTUs that always show up in the samples, versus yellow or green, where we have less frequent presence of species. So that kind of gets into the hypothesis of a core versus peripheral, and also the incidence of species in our seed samples. And I think with that, I'll close and open it up for any discussion.
Okay, do we have any questions for Dr. Little? I see none in the chat at this point in time. Well, I think you must have covered it very completely. Oh, there's one. Here we go. Why use the nested PCR approach?
Yeah, so this is an approach that we followed from another author, where they were using this sort of enrichment approach to kind of improve their targeted sequencing. Yeah.
Okay, and another question here. Do you have any idea if these microbes interact with each other?
No, maybe Ryan can help.
Uh-huh.
Well, actually, we've done a lot of in vitro studies to look at interactions, because we do think that some of the pathogenic species interact with some of the non-pathogenic species, and we don't understand how that's working. I view the seed as an ecosystem. I view the seed as its own little microcosm, and so I know that there is antagonism going on. I know there is competition going on, which is one of the reasons why when we do plating studies, we see some species that come out, and we never see other species that we might identify in the metabarcoding or metagenomics approach. Just because the species that grow out of the seed on culture generally are able to outcompete some of the other species that don't grow as quickly or are not as competitive. So that's sort of something that we can see, but it needs to be tested in better ways. So certainly I think that's a very complex situation.
Great. All right. Well, thank you very much. That was quite interesting, and we're going to keep moving it along. We've got Dr. Matthew Kirk, who's an associate professor in geology, up next.
All right.
There we go.
Let's see, share my video. There we go. Okay, hello everybody. Well, thank you for organizing the webinar, and thank you to those in attendance. One of the things that I'm primarily interested in as a researcher is groundwater geochemistry. And if you're interested in the chemistry of natural waters or aqueous systems, you really need to be interested in microbiology, because microbes drive a lot of the chemical reactions that occur in aqueous systems, both directly and indirectly. They have some really important impacts and interactions with chemistry that are not only very interesting, but also of great practical value. The research that I want to talk about today takes a top-down look at groundwater microbiology. Some of the work that we've been doing over the past few years is trying to better understand the impacts of agricultural land use on the composition of groundwater microbial communities. Advance, please. So it's well established that land use alters the chemistry and microbiology of soils in an agricultural setting. You know, depending on how you use the land, you've got different animal inputs, you've got different chemical applications, you've got different plants living on the surface, and you might be, you know, pumping water over that surface, or you're affecting the rate at which water is moving through there and flushing out solutes. So these changes, you know, these differences, let's say, in how we treat the land have a pretty big impact on the chemistry and microbiology of those environments. Advance. It's also well established that land use alters groundwater chemistry. You know, those changes in soils, right? That's going to affect the chemistry of water that's coming down through those soils and ultimately down below the water table and entering aquifers. You're also, you know, changing the cycling of water by pumping and flushing water through soils in the unsaturated zone. This is well established. You know, a good example of this is nitrate contamination in groundwater beneath fields used to grow crops as a result of fertilizer application. That's been known since at least the 70s. What isn't well known is how groundwater microorganisms respond to these differences. We don't know much about how land use affects groundwater microbiology, and that's one of the knowledge gaps that we've been chasing the past few years in my research group. Advance, please. So specific research questions that some of my students have been taking on the past couple of years include: how does land use affect groundwater quality? Does groundwater microbiology vary with land use? And does variation in groundwater microbiology mirror that in overlying soils? So the two students who led the research that I'm talking about today are shown here on this slide. It's Allie Lane on the left. She finished her master's in geology at K-State in 2018, and she took on the first two questions there, so she was focused on groundwater. And then Christina Richardson graduated in 2019 with a master's in geology, and she was working primarily on the second two questions. But they were both really great students to have the opportunity to work with. Next, please. So their field area was the Great Bend Prairie Aquifer in south central Kansas. This is part of the High Plains Aquifer system, so it's an extremely important water resource for Kansas as well as many other states. So in 2016, we went out and we sampled groundwater from the well nests that are indicated by the orange dots shown in this map. And we analyzed those samples for chemistry and microbial communities. And then in 2018, we went back and we sampled the soils around those wells in areas of different land uses. Next. One of the things that we found is that nitrate concentrations have increased in that groundwater, particularly in shallow wells with crops, surrounded by crops. So the box plot on the left here shows nitrate concentrations measured in 2016 from shallow portions of the aquifer and deeper portions of the aquifer in areas overlain by crops and pastures. And you can see that the nitrate concentrations in those shallow crop area from wells that are shallow and in crop areas, they really stand out. The standard for drinking water is 10 milligrams per liter of nitrate as nitrogen. You can see most of those samples have concentrations that are well in excess of that. So nitrate nitrate is a growing water quality issue in the Great Bend Prairie aquifer. Now these wells have been sampled after they were installed in the late 70s and 80s. And so we had a data set that we could compare our results to see how groundwater chemistry had changed. There were a lot of changes in chemistry, but in terms of land use, one of the biggest changes was nitrate. And again, we could, those shallow wells and crop areas really stand out. But it's also important to note that, nitrate's increasing throughout that aquifer. Groundwater moves laterally. So these changes, you know, they're going to be spreading out, these impacts of land use. Next. Okay, when we look at soils, we see a similar relationship. So these plots show nitrate, phosphate, calcium, and sodium concentrations in soil samples that we collected around those wells. For non-irrigated crop soil, irrigated crop soil, one site was a restored grassland. It was only changed in the past couple of years, and then pasture site. So the key comparison is really looking at these crop soils and comparing them to that pasture site. You can see that for all of those parameters, crop soils tended to be higher concentration, or at least range to higher concentrations. And you know, that's probably the result of chemical applications to those soils. And that's particularly true in these irrigated crops. You see the highest values there. One thing I forgot to mention on the previous slide was that the nitrate in the Great Bend Prairie aquifer, it's also coming from chemical application fertilizers. We analyzed the oxygen and nitrogen stable isotopes of the nitrate. We were able to demonstrate that it was largely from fertilizer application. So in terms of the soil and the groundwater, we're seeing changes in chemistry that reflect, you know, what we're doing at the surface. It's not that surprising, right? But at the end of the day, those changes in chemistry can have pretty big impacts on what's going on in terms of microbiology, who's there and what they're doing. Next slide, please. When we look at the groundwater microbial communities, we see that there is significant relationship between their composition and what is being done at the surface. What's the land use or management strategy at the surface? We don't have as much data as we'd like here. This is a preliminary data set. And I will be the first to admit that the groupings that you see here aren't as clear as you'd like them to be. But when we do the stats, we see that there is a significant relationship between land use and microbial community composition. One thing I should mention, this is a non-metric dimensional scaling plot of Bray-Curtis dissimilarities. So basically, the way to interpret it is the closer the points are to one another, the more similar those communities are to one another. So some communities that kind of group up based upon land use, well, that tells you that that land use is helping structure those communities. One thing that probably competes with the effect of land use in this aquifer is salinity. The groundwater gets more saline as you increase with depth very quickly. And in fact, we saw a stronger relationship between microbial community composition and well depth than we did between community composition and land use in our data. But so you got two kind of competing things that are influencing the microbial community, but it is fair to say that there is an influence of land use. Next slide, please. Soil communities, you've got, you know, much clearer distinction, or much clearer grouping by land use. It helps to have more data. More data are good. Part of the reason why we have more data is because we have more funds for sequencing at this point for this project. And it's also easier to collect soil samples than it is to collect groundwater samples, good quality groundwater samples. But so we see a stronger relationship there. You know, another thing that is also going to affect the extent to which these communities group up, in groundwater, as I mentioned before, the groundwater moves laterally. So it's a little bit easier to pin land use to a soil sample than it is to a groundwater sample. Some of that groundwater that you pump up out of beneath a crop field, some of that probably may have recharged under a pasture and so forth. Okay, but bottom line, we do see relationships that are significant between microbial community composition and land use in both soils and groundwater, which is pretty interesting. Next slide, please. When we compare the communities to one another, we see that there's quite a bit of difference between them. That's not very surprising considering that, you know, there's a different sample matrix here. Groundwater, we filtered microbes out of the cell, we filtered microbes out of the water. And here we're looking at a soil sample, so a solid matrix. So that in itself is going to introduce some differences in community composition. When we look at the taxonomy of those communities, there aren't like obvious common threads in terms of defining the response to different land uses. So we need to do a little bit more. There's some more work that we can do with these data to kind of try to find those relationships. One thing that is interesting though is in soils, we see that the soil microbial communities that plot nearest to groundwater communities tend to be the ones that have the highest nitrate concentrations. They also tended to be from irrigated crop soil. And irrigation water is groundwater, and it's going to contain some microbes. So you're basically spraying that soil with groundwater microbes during the summer. So that's a dispersal vector that's present for irrigated crop soil that's not there for pastures, for example, and we think would likely help create more of a common relationships between irrigated crop soil and groundwater than you would see in a non-irrigated field or pasture. Next slide. So the main conclusions, groundwater and soil chemistry do in fact vary with land use, and we see the same thing with the microbial communities. Again, this is a preliminary data set, but one I think that adds some support for seeking additional funding for research in these areas. Does variation in groundwater microbiology mirror that in overlying soils? I'm going to go with a very firm maybe on that. We got some more work to do on that question, but again, I think it would definitely be benefited by some more funds to try to take that question on. Next slide, please. So I'm very grateful for the people that contributed to this work. I mentioned Christina and Ali earlier. I also want to specifically call out Lydia Zeglin, who welcomed Christina Richardson into her lab for the soil microbial analysis. Very grateful for that, as well as contributions from other folks and the funding. And I thank you for your attention.
Great, thank you very much, Matt. I think we've got time for a question, if there are any burning questions out there. Seeing none, we will probably move on to the next. Again, I want to remind everybody that as you think through and have the opportunity to go back and look at some of at these presentations, should you choose to, feel free to reach out to these faculty members to get those links in place and answer questions and discuss from there. Great. Well, our next presenter is Dr. Sunny Lee. He's an assistant professor in biology. And Sunny, I will hand this over to you.
Thank you, Maureen. Hi, everyone. This project on antibiotics and how they affect or impact gut microbial community and function in early life illustrates our lab and how we use the information from amplicon data or marker genes to inform us on what kind of samples we use for metagenome shotgun sequencing and what we did with these shotgun sequences. This presentation only shows a very small portion of our results. But what I hope you can get away from my talk today is that one, the potential of using different types of methods to analyze the microbiome, and two, moving beyond just the identity of the microbial community to the microbial function. Slide. Next slide, please. So inflammatory bowel disease, or IBD for short, is emerging frequency. It's emerging with alarming frequency, both in the United States and around the world. Studies show that the early life events can influence the risk of IBD, which led us to our overarching questions in this study. Can we modulate the gut microbiome during the early life events? to reduce the risk of IBD. Slide. Multiple factors, including the gut microbes, are thought to be involved in the development of IBD. The environmental factors, diet, food, genetic background of the host, and also immune systems. In our lab, slide, we study the pathogenesis of IBD in the context of the gut microbiota. Slide. Several recent studies present an association between the early life exposure to antibiotics with increased risk for IBD development. And with that overarching goal in our mind, our first objective is to determine if early antibiotics exposure plays a causative role in IBD to better understand this IBD pathogenesis, and also to the ability to develop therapeutic strategies. We use a mouse model, IBD mouse model, which allow us to provide more advanced insights into this host microbe interaction in the context of the IBD disease. We treated half of the dams with broad-based antibiotics during peripartum and examined the offspring of the gut microbiome using fecal samples on 3 time points at 3 weeks, 7 weeks, and 11 weeks of age. Slide. This figure demonstrates the amplicon sequence variance, or ASV for short, relative abundance. Each of these columns represents a single ASV or bacterial population. Each of these rows represents a single fecal DNA sample. The heat map reveals one very distinct pattern is persistent differences between the control, which is marked by NT, and the antibiotics treated, which is marked by CPZ on the extreme right column, from 3 weeks of age through the 11 weeks of age. In addition, we can point out 2 main characteristics of this treatment. Slide. First, the most commensal firmicates indicated by this red box survived the peripartum antibiotics treatment. 2, slide. Most of the commensal bacteroides shown in this green box were lost after CPZ treatment or the antibiotics treatment. And finally, 3, slide. Some of the bacteria, which is really interestingly shown in this pink box belonging to Bacteroides and Verrico microbial, re-emerge at 11 weeks of age. In addition to what you see here, the diversity of the antibiotics group was much lower than that of the control group at 3 weeks, 7 weeks, and 11 weeks of age. And this biopsies at 3 weeks of age is thought, or we hypothesized it to be vertically transmitted from their dam. Overall, these findings demonstrate that maternal peripartum antibiotics exposure induces persistent dysbiosis in their offspring. Slide. So using the results from the 16S amplicon sequencing, we did shotgun sequencing on a subset of samples. Looking from the left to the right of the slide, each column represents a metagenome assembled genome, or MAX for short, or in some sense you can consider them as bacterial populations. And each row represents a fecal sample. On the extreme right of the slide, there are different characteristics such as age, sex, and presence of colitis of these samples. Slide. Paying attention to just a small subset of these MAX in this blue box, looking at the samples on the very, very top of the blue box, you can see that these MAX are present in the control dam, but absent in the dam that are treated with antibiotics. Moving down this box towards the middle samples, similar to those MAX, similarly these MAX were absent in their offspring of the dam that were treated with antibiotics. However, what is more interesting in this is that at week 11, looking at the most bottom part of the blue box, you can see that these MAX actually reappear in the offspring, and it coincides with the appearance of the colitis in these mice. we were curious about why this bacterial population, which was lost during the antibiotics treatment, and then it caused colitis in the offsprings when they show up in the offsprings during later parts of their life. Next slide. So next, we look at the bacterial community as a whole, rather than a single MAC or a single bacterial population. And one of the important rationale for doing this whole community analysis is that the host itself may not care about individual microbes, but rather what is the resultant product from an entire microbial population. Slide. Next slide, please. Looking at the entire bacterial population, this, I think we went too much. Can we have one slide back, please? Yes, thank you. Looking at the entire bacterial population, this figure represents the microbial functional potential of each sample during the 3 weeks, 7 weeks, and 11 weeks of age of the offspring. The red dots represents offspring where the dam is treated with antibiotics, and green are the controls. It is consistent with the results that we got from 16S amplicon as well as the MAX analysis. We notice that there is a persistent functional difference in the gut microbiome between the two treatments during the 3, 7, and 11 weeks of age. Next slide, please. However, we also noticed that only a subset of the offspring that was treated with antibiotics developed colitis. So we zoom into this group of offspring that dam was treated with antibiotics. Looking at this group of offsprings, the red dots are the offsprings that develop colitis, and the green dots are those that did not develop colitis. We notice that there's only a functional differences between the microbiome only at 11 weeks of age, but not at week 3 or 7. And one of the significant functional difference between the colitis and non-colitis group is nitrogen metabolism. We went back to the MAX and realized that the MAX that show up at 11 weeks of age and correlate to the colitis in the offspring also has a higher relative abundance of genes related to nitrogen metabolism. Slide. So the basic conclusion from looking at the microbiome in these 3 views, from 16S amplicon to MAX to the whole community, is 1. Antibiotics usage during peripartum resulted in persistent dysbiosis and impaired immune development in the offsprings. And this will result in a risk factor for IBD. Moving forward, we think that there are certain bacterial populations that are essential for the host immune education in their early life to maintain health later in their development. Slide. From this study and synthesis of all other publications regarding this IBD development, we think that nitrogen metabolism could be one of the important microbial functions that can influence the risk of IBD in the development of a host. We are nowhere near finding the therapies for IBD with this study, but we are definitely taking one more step closer to the results that we are hoping for. Slide. I'd like to thank my talented lab members and collaborators, and this study is supported by this grant, and thank you. I'm open for taking any questions.
Great, thank you very much. That is some fascinating work that you've gotten started there. Are there any questions from the group for Dr. Lee? Silence. Just a question from my own curiosity. I'm not sure that this is even going to come out well, because I'm looking at it strictly from a lay person, looking at the kinds of work you're doing. What would be a timeline in coming up with a better understanding of what's happening in the gut or in the body with IBD issues. Do you see this as something that's going to be taking several years? Or is that even a fair question?
I think it depends on our ability to interpret. At the present moment, I think we have more data than we need. Without doing any research at all, I think we have enough data. If we are just doing data mining, it will take us at least 5 to 10 years just to get a glimpse of what is happening in the gut. We are limited by our knowledge of the microbes and not by the amount of data we have. So if you ask me this question, I would say that if we can move our knowledge fast enough, we are looking at about maybe 8 to 10 years.
Fascinating. There is a question that just came in. What is your opinion of the whole microbiome therapy of fecal transplant therapy?
Whole microbiome. So one of the issues with fecal transplant therapy is that we do not really have a knowledge of what is being transplanted. A lot of times in terms of fecal transplants, in order to make it more palatable, we try to not transplant the fecal material, but put it in a pill. That's what a lot of technology is. But in order to put viable bacteria in the pill, most of these bacteria are actually spores. However, in terms of transplanting spores, it might not be successful in colonizing the new gut. Also, in terms of fecal transplant, there is a lot of unknowns, and one of the unknowns is transplanting pathogens. There have been, I think just a couple of years ago, there was a couple of deaths resulting in transplanting of pathogens to another patient, and there are always risks in there, and I think we need better screening of the host and better screening of this material, this fecal material, before fecal transplant therapy could be more in terms of promoted.
Very good. That's a good question. Okay, are there any other questions for Dr. Lee? No? Great, thank you so much. Thank you so much. That was really very interesting. The next presenter that we've got here is Dr. Megan Niederwerder. She is an assistant professor in veterinary medicine in the virology area. So Dr. Niederwerder, take this one.
Well, good afternoon, everyone, and thanks so much for Courtney and Maureen for organizing this meeting and for all your work in the global food systems. It's been fascinating so far. I've really enjoyed hearing from Doug and the other members of our K-State faculty that are really showing the diversity of the microbiome research going on campus. So the talk that I'm going to give is some of the research that we've been doing in my laboratory, looking at how can we utilize or understand more about the gut microbes in the context of improving swine respiratory disease. Next slide. So when we think about the definition of the microbiome, Joshua Lederberg really coined this term or coined this definition early on as the ecological community of commensal, symbiotic, and pathogenic microorganisms that literally share our body space. Please advance. Oh, there we go. So we can simplify that a little bit even more to think about the symbiotic organisms, or those that provide us with benefits, and those pathogenic microorganisms that cause our bodies harm. And so I'm a veterinarian, I'm a virologist, and really the focus of diseases and infectious diseases have been focused on those pathogens and how do we eliminate the pathogens, either through antimicrobials, if they're bacteria, vaccines, if they're viruses, and those symbiotic organisms have been really largely ignored in our history. And so when we think about the microbiome space and what we're trying to do with pigs, please advance, is that the focus shifts. It's really a paradigm shift away from the pathogen and more on those symbiotic organisms, and that do we have the capability to increase those symbiotic organisms and actually outweigh what the pathogen does. So let's say we have an endemic disease that's been around for many decades. We've tried many different routes to eliminate that pathogen, and we haven't been successful. As opposed to focused on that pathogen, can we actually focus on those beneficial organisms and again outweigh the effects of the pathogen on the actual host health? Next slide. So all my work has been done in pigs. So we're looking at swine health. And when we think about what impacts pig health and the microbiome development of the pig, even within the first 3 weeks of life, we can see that there are quite a few characteristics or factors which may impact microbiome development. So when we first think about this, the vast majority of piglets are born through the vaginal canal of either sows or gilts. That's really their initial microbial exposure. So as they're going through the vaginal canal, they're ingesting microbes. That, of course, can play a role in their microbiome development. Then they live with the sow or gilt for approximately 3 weeks as they nurse on their mother. Of course, the microbiome of the sow or the gilt, as well as the diet of the sow or the gilt, can really significantly impact the microbiome development of that piglet. We also know that there are environmental factors, such as are the pigs housed indoors versus outdoors, are they housed on dirt versus slatted concrete floors, can really impact that microbiome development and, of course, the diversity of the microbes by which they are exposed. Most of our commercial pigs are actually housed indoors, and even some of them live in filtered barns where all the air that comes into the barn is filtered. So you can imagine there can be this significant variation with regards to the microbes these piglets are exposed to in the first several weeks of life. We also know that the diet of the piglet, of course, pigs are typically weaned at 3 weeks of age and then transition to solid feed. That can impact the microbiome development, handling and stress, as well as injectable antimicrobials, pathogen exposure, as you can imagine, anything that causes diarrhea, but also other systemic diseases can impact the gut microbes and in-feed antibiotic growth promoters. I like to show this slide initially because, as you can see, there are multiple opportunities for us to intervene and either provide beneficial effects to the gut microbes of these piglets or detrimental effects of to the microbiome of these piglets, again, even with just those first several weeks of life. Next slide. So when we think about the gut microbiome and respiratory infections, one of the big questions we get is, why are we talking about respiratory disease? Infectious respiratory disease is really a leading cause of morbidity, mortality, and antimicrobial use in swine. And it's also a leading cause of morbidity and mortality, of course, in neonatal humans. And when we think about these respiratory diseases, again, many of them have been around for many decades. Maybe we don't have a perfect vaccine. And so we look at the gut-lung axis as sort of an alternative tool to improve the health of animals that maybe have been exposed to respiratory pathogens. So the gut-lung axis is really what we call the bidirectional communication of how respiratory diseases can impact the gut microbes. And of course, the metabolic products of the gut microbes can impact the immune system that is primarily focused in the lung. And so we know there's this communication between those two main organs. Next slide. And in fact, when we take this a step further to look at what's been published in the literature, you can see on the left-hand side of your slide, there's actually been several respiratory pathogens that have been investigated primarily in mice models with looking at their associations with outcome and the gut microbiome characteristics. So you can see there are viral respiratory pathogens such as influenza virus, bacterial respiratory pathogens such as pneumococcal pneumonia, and even fungal respiratory pathogens. And what's interesting in that center column is that oftentimes the microbiome characteristics in the gut that are associated with improved respiratory outcomes are very similar. So oftentimes we see the presence of endogenous microbiota versus a mice model that's been treated with antimicrobials. having the presence of endogenous microbiota oftentimes improves the outcome of that mouse after they've been exposed to the respiratory pathogen. We also see things like fecal microbiota transplantation, increased microbial diversity. These are fairly consistent factors with regards to microbiome characteristics that seem to improve respiratory health. Next slide. So thinking about what we know with human infectious respiratory pathogens, the impact on weight gain and immunity, the focus of the work in my lab has been to look at how this translates into a porcine respiratory disease complex model. Next slide. To simulate porcine respiratory disease complex, we actually used two viruses, porcine reproductive and respiratory syndrome virus, or PRRS. This is actually what's considered as the most costly disease to swine production in the US. So this, as the name indicates, causes respiratory disease in those young and weaned pigs. And we also use a second virus, porcine circovirus type 2, as the viruses, when combined, actually increase the severity of the respiratory disease, but also work together to reduce weight gain as well as cause immunosuppression. Again, simulating a polymicrobial respiratory disease complex that we would see in commercial field situations. Next slide. We also use the Lawrence Livermore Microbial Detection Array as what we have to historically utilize to characterize the microbiome. This is a microbial detection array. It's a pan-microbial array. It allows us to detect over 10,000 microorganisms in a single test that includes viruses, bacteria, archaea, fungi, and protozoa. Next slide. So the first questions that we had in my laboratory was really just to understand, are there microbiome characteristics that are associated with outcome after pigs are co-infected with PRRS and PCV2? And you can see on the top part of the slide, publication, our first publication was looking at what are the microbiome characteristics at 70 days post-infection when we compare those pigs that had the best clinical outcome, or maybe those pigs that didn't have any clinical signs, and compared those to the worst clinical outcome pigs, or those that had severe clinical signs. And our initial studies found that microbial diversity, as well as the presence of a non-pathogenic E. coli, was associated with improved clinical outcome, again, in pigs 70 days post-infection. We took this a step further in the publication that's on the lower half of the screen to understand were there microbiome characteristics prior to co-infection that actually predisposed or predicted outcome. And we found that several characteristics, including, again, increased microbiome diversity, as well as the presence of streptococcus and ruminococcus when present prior to co-infection, actually improved subsequent outcome. Next slide. So this led us to the question, can we actually modulate the microbiome prior to co-infection to improve subsequent outcome? And when we thought about how we could modulate the microbiome, we just talked about this, we looked at fecal microbiota transplantation, or FMT. This is the process by which feces are donated from a healthy donor and subsequently transplanted into a diseased or young individual. The way which FMT is efficacious for the vast majority of diseases in which it's been studied, the way in which it provides that efficacy is largely unknown. But we think that the way FMT provides those benefits is through increasing microbial diversity, enhancing those beneficial microbes, and then modulating the immune response. Next slide. So we looked at this with a 20 nursery-age pigs and looked at the effects of fecal microbiota transplantation in the fecal group seen on the lower half of the screen, compared that to a saline mock transplant. This fecal microbial transplant was collected from high health donor sows that had several characteristics of high growth litters and healthy litters. The transplant was administered orally for 7 consecutive days prior to all pigs being challenged with PRRS and PCV2, and then monitored for 42 days post-infection. Next slide. What we found in the transplanted pigs, which is shown in the black squares, compared to the control pig, which is shown in the white squares, we found that the transplant actually had a protective effect against morbidity, or those pigs that showed clinical signs, and actually resulted in the transplanted group receiving 60% fewer antimicrobials compared to the control group. We also saw a protective effect with regards to the transplantation in the overall mortality rate of the pigs, as well as the consistency with regards to weight gain, which is shown on that bottom half of the screen. The transplant actually provided a protective benefit and reduced mortality by 70%. Next slide. We also looked at lung lesions, showing that the transplant actually reduced the lung lesions in those pigs, as well as enhanced antibody production in those transplanted pigs. So you can see both PRRS and PCV2 antibody on the right side of the screen. We saw an enhanced immune response in those pigs that had received the transplant compared again to those mock saline controls. Next slide. Oh shoot, this got messed up. But overall, what I want to show here is that that's okay. The majority of the work that we've done so far over the last several years has identified several clinical health characteristics, such as on the bottom side of the screen, such as increased antibodies, reduced interstitial pneumonia, increased average daily gain, and associated those with several gut microbiome characteristics, such as fecal transplantation, increased microbial diversity, increased ruminococcus. So we're really starting to identify some of these characteristics that, again, predispose or predict improved health outcomes in pigs when exposed to respiratory challenge. Next slide. And overall, with the goal of utilizing the gut microbes as an alternative tool for these porcine respiratory diseases, again, when traditional strategies have not proven to, of course, completely protect the pigs from these diseases. Next slide. And these are several of the collaborators as well as the funding agencies that have supported this work. And I'd be happy to take any questions at this time.
Thank you so much, Dr. Niederwerder. I do see one question here. Were all of the, let's see, nope, there are two of them. Any ideas concerning the recent COVID-19 disease and human gut microbiomes, given your experience with the porcine microbiome and their respiratory viruses?
Yeah, you know, I've thought about that, because I think that when we talk about things like the germ theory in humans, where we're all being exposed to less microbes early on in life, oftentimes our neonatal infants are severely affected by respiratory syncytial virus and other respiratory diseases. We, of course, I would presume and assume that the gut microbes in humans right now is impacting outcome with SARS coronavirus 2. And I think that will be a very interesting question that hopefully comes out of future research, looking at are different populations of humans differentially affected by their microbiome characteristics. Of course, we know this is impacted significantly by developed versus developing countries, diets, environmental conditions for humans. And so I think that will be an interesting question for the future. But of course, anybody exposed in the microbiome world will go, it's probably affecting outcome. Okay.
Another, oh boy, you've got several of them coming on. I'm not sure we're going to get to all of them here this afternoon, but we'll do our best. Were all of the hogs with increased microbiome diversity of the same genetic line? If not, were there any differences between genetic lines?
So we get all of our pigs from, when in any of these studies, the pigs come from the same genetic source. They come from the same herd, the same even barn. Because we think that, of course, that there's a lot of differentiating factors with regards to environment, genetics, and how those microbes actually colonize. The other thing I do with regards to the control versus the transplanted group is that those pigs are actually brothers. that are separated into two different groups so that, again, I can somewhat control for genetic variation.
Okay. Could you determine the microbiomes of the control and FMT treated swines? Will it help determine if any beneficial groups are enriching or their immune modulations?
Yeah, that's been a big focus because what Sunny said is true in that when we think about fecal microbial transplantation, one thing is we don't expect everything to colonize. Some of those microorganisms may colonize permanently. Some of them may have some sort of immune stimulus and an interaction with the gut lumen for a short period or maybe for a longer period. We're transplanting cells into the, colonocytes into the new piglets. And so, yes, absolutely. One of the big factors is after you do the transplantation, what actually colonizes, what stimulates the immune system, and can we isolate that to, let's say, a handful of microbes as opposed to a whole community, which is what we would transplant with FMT.
Okay, one last question we'll get to, and then Dr. Gare will definitely tack on a few minutes on your presentation here. But the last one is, could you elaborate on the fecal transplant? What specifically is in it, and what is the source, and how reproducible is it?
Sure. So we collected feces from 2 sows that were in a herd that was negative for PRRS and mycoplasma and several other pathogens of swine. We took the feces and actually processed it based on a protocol that's been published in the human literature. I think it's a 2012 Hamilton publication. Essentially, you mix the feces in with saline, you homogenize it, you centrifuge it, you concentrate the microbes, and then you add glycerol and freeze it so that you can utilize it at any time. And we have characterized the fecal transplant. The microarray actually detected, I think, over 60 individual species, of course, several phyla, and we have characterized that as well as with sequencing.
Great. Okay. I will also state, since I've got a captive audience here, we completed a Global Food Systems podcast under the Something to Chew On title with Dr. Niederwerder recently, and we put it up, I don't know, it's probably been about a month ago, maybe not quite that long ago. So if anyone's interested in listening to more about the research that she's been doing, it was a very, very interesting podcast that we did. So thank you so much. Okay, we will move on now to the last but not least presentation of the day. Dr. Tara Gare is also with the College of Veterinary Medicine, and he is a research assistant in the pathobiology group. So, Dr. Gare.
Thank you. Good afternoon, everyone. Actually, I'm a graduate student doing PhD and working with Dr. Victoria Borko at the College of Veterinary Medicine here at K-State. And I'm presenting our work on the age dynamic of fecal microbiome and also the microbiome composition and antimicrobial resistance in production and breeder soil. So this work has done with collaboration with Dr. Tokas from the Department of Animal Science and Industry at our university, and also other collaborators from the College of Veterinary Medicine and Texas A&M University, and also the University of Missouri. Next slide, please. So we performed the longitudinal study of production pig and breeder sows cohort, and our main hypothesis of this study was age is the strongest driver or the main driver of fecal microbiome, as not only fecal microbiome, but as well as the fungal composition, we call it the microbiome composition, as well as the antimicrobial resistance, rather than the production system or environment in case of the farm animal host. So in this study, we elucidate and compare those dynamic, both fecal microbiome as well as microbiome composition and the antimicrobial resistance in cohort of production pig and breeder sows. Next slide, please. So we designed the study, longitudinal study. In the first, we follow the production pig, one cohort that with the 12 animal balanced by gender, And we follow this cohort from close to the birth, 2 days old, up to the day of harvest, which is 180 days or 6 months. Similarly, we follow the breeding sows, 2 cohort, first cohort 6 animals and second cohort 12 animals, from about 3 weeks old until the day of finishing or weaning of first pairing or parity. And in both cohort, we sample the 9 different time point, and we choose this time point because when they move to the next facility or diet change, or in case of breeding sows during the mating, or when they are transferring the pairing house, so that we can capture all the information during the production system. Next slide. So we collected the fecal sample from individual pig, and we store that fecal sample both for the metagenomic analysis and for the microbiological analysis. For the microbiological analysis, we store the fecal sample with the 50% glycerol. And from those fishes with the glycerol, we quantify the total and the antimicrobial resistant coliform enterococci by spiral plating. These coliform enterococci are the indicator bacteria. So similarly, for the metagenomic analysis, we extracted the community DNA from the fecal sample, and we performed the microbiome analysis, and as well as the fungal microbiome analysis. And we utilize the both methods, like earlier method, OTU-based methods, and now this has just moved to the another field, amplicon sequence variant. So we have a both. We utilize both approach to analyze those data. But now in this presentation, I will focus mainly on amplicon sequence variant data. And apart from that, we also analyze the antimicrobial genes in metagenomic using the shotgun metagenomic approach. So in this study, we aim to utilize both cultural method as well as the metagenomic approach to get the better picture of the AMR in the host. Next slide, please. So here we can see the fecal microbiome taxonomic composition is family level relative abundance. The in production pig in top graph, and the lower graph is from the breeding sows, and this red rectangular box actually showing the age overlapping between the 2 cohorts. So when we can, if we see those overlapping age period between the production cohort and breeding sows, actually the taxonomic composition looks similar. Next slide, please. So here we have a fecal microbiome taxonomic composition, again from the production pig and breeder sows. These microbiome taxonomic composition, they are not similar as a microbiome. For example, day 77 in production pig looks a little different, but overall the taxonomic composition looks similar between the production pig and breeder sows. Next slide, please. So now we look at the specific indices for the richness or diversity of both microbiome and microbiomes. So here we have a chow richness of the species. So in the right side, we have a fecal microbiome richness, and the left fecal microbiome richness. And so first things we notice that from the early age, actually the richness is increasing. So overall trend is increasing from the early age to the until the harvest. And when we compare the both cohort production and breeding sows, for example, from the day 22 from the production pig and until the 154 in the breeding sows, the overall trends look similar in both microbiome as well as the microbiomes. The richness of the species. Next slide, please. So here, in this graph, we are showing the diversity of the species. again, the diversity of both microbiomes actually increasing from the early ages to the until the harvest. And when we compare with the breeding sows within those overlapping age period, actually overall trend looks similar. As well as in the microbiomes, the overall trends looks also similar in both cohorts. Next slide, please. So now we evaluate the phenotypic AMR in the selective indicator bacteria from the microbial community. Here we estimate the bacterial density that grows in the presence of the antibiotics, in the specific antibiotics. So we choose the clinical breakpoint for the concentration of antibiotic in the media where these coliforms or enterococci grows. So here we can see that, so the top graph is from the production peak and lower is the breeding sows. And the actually coliform count that are resistant to the antimicrobial resistance to these drugs, actually it's appear to be highest in early ages and subsequently declined with the ages. And when we compare with the breeder sows, the overall dynamic of this coliform, that the resistance to these drugs looks almost similar in those overlapping age period. Next slide, please. So here we have another bacteria, enterococci, that are gram-positive bacteria, and these are the selective indicator bacteria from the microbial community. So here also we can see that in both production and the breeding sows, the that are enterococci that are resistant to these drugs actually appear higher in early ages and subsequent decline, and overall trend or dynamic looks similar or comparable between these production peaks and breeder sows. Next slide, please. So in addition to the phenotypic data or only on those specific indicator bacteria, we also did the metagenomic sequencing, shotgun sequencing to get a better picture. So for this, we choose, this is for the preliminary data. So we sequence 2 production peak for S point. And here we can see the sum of the gene copy within the AMR gene classes, for example, beta-lactam, fluoroquinolones, and tetracycline, and so on. And so, so looking at this result, we can see that the actual gene within that class appear in early ages and disappear and appear re-again in the like in finisher states. So, and some classes of antibiotic are present across all ages. So this color, the color bar represent the when the animal move to the different facility. So it's interesting to see that some of the antibiotic resistant genes are present all ages and disappear and appear again. So in this stack bar with the colorful stack bar, also we can see that the diversity antimicrobial genes in class diversity was actually highest in early ages, and then decline up to the weaning, then again increase. So next slide, please. So from this study, from the early age to young life, we saw that the age-dependent dynamic of richness and diversity of fecal microbiome as well as fungal microbiome taxonomic composition were similar or comparable among the production peak and breeder sows cohort. And the age-dependent dynamic of phenotypic AMR in fecal bacteria were also comparable among these cohorts. And we actually, results suggested that the highest diversity and abundance of the AMR gene occur at the earliest point and decrease with age. Next slide, please. So from this study, so it led to the another questions. Are So we saw the age-dependent dynamic in both microbiome as well as AMR. And this led to the another question that are these there are interaction between age-related dynamic and effect of diet and the antimicrobial treatment on the fecal microbiome or AMR. Because a lot of time we are that diet also affect the microbiome or microbiomes, or antibiotic treatment also affect the microbiome composition. So to address this question, we designed a study in production peak cohorts. So where they received the where the production peak cohort in each finisher farm, approximately from 93 days, they randomly allocate one of the 3 dietary treatment, and this dietary treatment was composition of this dietary treatment was based on the different level and source of fiber. So namely control and distilled dried grain with the soluble with the different level of the fiber, and which called DGGS, and the finally another diet is called beet pulp with the different level of fibers. So, so within this study, there is another sub-study, but mainly I will focus on the first part. So to answer this question, is diet also affect the microbiome or AMR? So we follow these pigs that are feeding one of the 3 dietary treatment from the 93 old to the until the day of harvest. Next slide, please. So again, this is the fecal microbiome taxonomy compositions from those production pig that is not treated, but get and receiving the different diet. So x-axis there is so you can see the age, and the on the top there is a different diet. So the composition, so stack bar is showing the microbiome compositions based on the diet. And the composition looks little variation among the diet. Next slide, please. So now,
excuse me, in the interest of time, I know we've got people that are going to start, that have started to drop off here. So if you would like to kind of pull this together and complete, that would be wonderful.
Okay, thank you.
Thank you.
We also compare the richness and diversity of microbiomes of this study, and we can see that the richness and diversity are actually similar in control and beet pulp diet, but in case of dried grain DGGS, actually there are some evidence of the age and diet interactions. Next slide, please. Similarly, with the phenotypic AMR in both coliform and enterococci bacteria, result suggesting that there is a age and diet interactions. Next slide, please. So the conclusion from this study, the dynamic of the taxonomic richness throughout the finisher phase were similar in pigs on the corn-based diet, which is control diet, standard production diet, and beet pulp-based diet, but there was interaction between the age and DGGS diet affect the richness on microbiome taxonomy composition. And across the 3 diet, there was interaction between age and diet effect on diversity and evenness of taxonomic composition. And for the antimicrobial resistant data suggesting that interaction between age and diet effect on fecal bacteria, but we are doing the statistical analysis for that. Next slide. So with that, I conclude my presentation, and I'd like to acknowledge for the funding and my advisory committee. Now I'll take the question.
Thank you very much. Are there any quick questions on this particular study? Quite interesting. I really appreciate it. I want to step back now and thank all of you for the time you provided in putting these studies and these presentations together. It was fascinating, and it's just amazing the amount of work that's happening across campus. I'm hoping that maybe we made a couple of connections during this workshop, and that whether it's virtual or hopefully soon face-to-face, you'll be able to meet with some of your colleagues and continue the discussion in this area. I want to remind all of you that are on to please fill out the review forms. Those are very, very helpful to us. And again, thank you for giving us your time. I know that time is critical at this point, and it's pretty tough to come by when you're working from home with all of the distractions that Peter had mentioned earlier. So with that, thank you for attending, and I hope to have the opportunity of seeing you all soon. All the best.